I'd like to thank all the speakers for keeping a great time, so we'll have a little bit of time for discussion. So just some summary slides. So I think as you've seen across the 4 presentations, this type of work, even though overall the topics are similar, trying to look at where parasites are coming from and how they're moving, it's really not one size fits all. It depends on the spatial and temporal scale of the question that you're asking, and also the underlying population of parasites, which is going to differ from one area to the other. So the key questions are what is a research question or set of questions that you're really interested in? What is the overall endemicity of the setting? And I think in addition to endemicity, what are the patterns of local versus importation, which I think will also affect the underlying parasite population and its stability. Which markers do you want to genotype, both in terms of getting the information that you want and trying to balance the cost, the complexity, the speed at which you can get these data, how comparable they are from one lab to another. And then finally, something we didn't discuss in great detail, but is probably one of the most important things, is what is your sampling plan? If you're trying to look at transmission within a village, you really need to sample probably lots of people within that village. If you're trying to look at connectivity in a set of different regions, you need to make sure you have samples enough from those different areas. And a lot of times, a lot of the work that I think a number of us have presented has been opportunistic, based on things that have been collected from other studies. And prospectively, if we have important questions to ask, I think we really need to target the sampling. And I think there's probably really efficient ways we can do that. Validation of new marker panels is really important. I think Alyssa presented some data to suggest that you need to not only identify things specific for your area, but then really kind of rigorously validate them in separate sample sets. And then some of the other considerations: budget, resources, time. So some of the discussion points that we came up with on our own, but really interested to hear if there's other things that the audience is interested in, are there particular approaches that can be done universally, or does everything have to be done locally? If you're not doing a whole genome sequence, is there some crossover between those two? Is there a minimum set of markers for a comparison of different data sets? It's a big question, but maybe we have some thoughts on that. And then I think, in addition to other things from the audience, I think something that was brought up during one of the talks was, what is the turnaround time, and how does that influence the utility of these data for programmatic use, and how quickly do we need to get these data, and what are the barriers to getting these data in a timely fashion? Any thoughts on what you want to start with? Anything from the audience that people found particularly salient and would like to discuss, either amongst folks in the audience or for panel members?
Very nice talks, all of you. So, Bryan, I think you said you can genotype samples with less than 10 parasites per microliter. And just one thought I had is, I think we see now emerging from mosquito feeding assays that these very low density infections are really very unlikely to be taken up by a mosquito, both for Vivax and falciparum. And as a result, it would be much less likely that you really would see 2 people having the same parasite, because it will not be transmitted. So have you, or I think Ananias touched on it when he mentioned that the clinical samples were more closely related than the asymptomatic. Have you ever really essentially integrated parasite density and the chance that actually an infection is taken up by a mosquito and transmitted in the surroundings? Have you ever integrated this somehow in your studies?
That's a great question. So we haven't explicitly modeled that yet. I think it's an important question. I think there's a lot of factors. So of course, if you have a low parasite density at one point in time, it's quite possible that 2 months ago, especially if it's an asymptomatic infection, or 3 months ago, you had a much higher parasite density that could have been transmitted. And it can also prospectively go up. But you're right that overall there's a lower probability of being transmitted. But we've seen, maybe this partially answers your question, we've seen in the studies that we've been doing in Swaziland, where we have asymptomatic and symptomatic populations, that we do see more onward transmission potential from the asymptomatics. And it may not be at that point in time, but perhaps over the last few months, because they've had that infection for much longer. And a number of modeling studies by Toon Bausema and Chris Drakely have shown that in certain communities, the vast majority, even though the transmission potential of any given individual is higher for higher parasite densities, more transmission might be actually occurring in asymptomatic and lower parasite density infections. I think there's also a third issue, which is a technical issue, and that is when you have low parasite density infections, it's probably much harder to detect all of the different clones that are present there. And many of these infections are, if anything, more polyclonal because these tend to be people that are not seeking care, not getting treated, and having more infections stacking up.
I think there are other two problems there. One, what you call the infectivity of the vector, I think is also vector specific. I don't think the requirements for Gambi to get infected are the same requirements than Darlingi or RB manus, for example. And I think that's something that need to be, addressed. And the other issue is when we talk about the efficiency of asymptomatics, we have to understand something. It's different. You may be, let's say, 5% efficient compared with asymptomatic. But if you are asymptomatic for a long period of time, that 5% start to add up. Okay? So having this, how long are you asymptomatic and participating in transmission is important, and that's something that we really don't have a good grasp of that yet.
So this has been a great session. I just want to say that I think the point of local versus universal approaches is a really salient one for the field to think about. I think if cost and practicality weren't an issue, we would all love to work with the whole genome data, and that would be ideal. But of course, we've heard about different SNP panels using different genotyping approaches and microsatellites as well, and they all have their advantages. If we use local approaches, if, for example, we pick a set of SNPs to use because in a particular sample set it helps us differentiate between populations A and B, then we have to wonder if we apply it to populations B and C, if the ascertainment bias is affecting our estimate of relative divergence or connectivity. So I just want to underscore that, but maybe kind of referencing Shannon's talk just raised the notion that perhaps IBD and similar tools are a way of sort of transcending specific markers that might comprise panels and kind of translate these analyses to the level of thinking about haplotypes or genomic segments that are a product of recent common ancestry, as opposed to analyses on the markers themselves and minor allele frequency differences in FST and things of that sort. So just putting that out for discussion. But
thanks, Dan. That's a great point. So what sort of data would you use the IBD on whole genome sequence data? Or?
So at the risk of referencing Amy's paper one more time in the session, I think it could work on whole genome data that might come from selectively whole genome samples, but I think it can also work, as Amy showed, on panels of 100 SNPs. I think with 24 SNPs, it's tough to meaningfully infer IBD. At least across the whole genome, but with panels of 100 SNPs, there's evidence that it works decently well.
Yeah.
So.
Dan, do you think that would work in? I'm just going to keep you out there. Do you think that would work in areas of higher recombination, like sub-Saharan Africa?
I'm not sure it's a universal approach yet, Bryan, in that sense. But I think that would be, depending on the samples that you have. I mean, this is a point you raised. If you have samples collected with sufficient density within a very local area, maybe you would have enough IBD to even detect meaningful signals in parts of sub-Saharan Africa with really high transmission. I think that remains to be seen. I haven't seen a whole genome sequenced sample set at that resolution, or at least I'm not aware of it. Olivo may have something to add.
I don't know. I would say that with the 100 SNPs, which is what we work with, there will be special cases where you might be able to infer IBDs, but that wouldn't be the general rule, I would say.
Is Amy here? There she is. She's Amy, do you want to comment on this issue?
Well, I think your initial focus is very... If you're focusing on the tail of the IBD distribution, and you're really looking at closely related siblings, then you can probably get away with a lower number of SNPs because you're then looking at larger blocks, basically. So, but it is, very dependent on where and in the local settings and how much recombination is happening.
I think a key point is that, you know, it is hard to have a universal approach unless we can do whole genome sequencing and cherry pick the SNPs we want for our local population. So that's an important point, I think, to make from this session. We're all using different approaches, and you know, what's the point? The research question is quite often what's going to apply to a control program, and that's often in a local or a regional setting. So, yeah. Yeah,
I mean, I guess one other kind of thing to consider is it takes a long time to develop these tailored local approaches. I mean, you've presented a great example of this, but it probably took a few years and some money to do that. And there might be a balance between developing a larger set of markers that incorporates numerous local panels, if you will, or tries to overlap that in some way that you get meaningful data from a lot of settings, and then you can augment that with other data. And I'm not really, haven't done really enough thinking about that mathematically. I think, what sets of markers that would be, but it might be an interesting exercise for the community to think about is, how can we balance these needs to get really focal data with kind of more global things that are standardized and comparable and don't require constantly redeveloping things, especially if we're talking about areas of low transmission where there's not going to be really stable populations of parasites, and that even the focal markers themselves might change over time if there's a lot of migration.
Yeah, this might be a naive question. It may be a question for Dan. It was just triggered by his suggestion that I hadn't thought of before, like the concept of possibly reporting everything in terms of IBD as a way of getting around the issue of whether you're using local or universal markers. But as I start to think about that, I'm not quite sure what that would look like. And it occurred to me there may be people in this room who've looked at modeling that. Certainly, if you were using thousands of markers, I can see how that would work. But with only hundreds, it seems to me like you'd probably wind up with very confusing differences in block size that you'd wind up with. I mean, is that going to be workable? I don't know if this is something that Amy has looked at or Dan. It could be pretty easily modeled based on available whole genome data.
Dan, do you want to answer that one?
Sure. I'll just say I think Olivo's sentiment is just that it's hard to estimate IBD indeed at high resolution from small panels of markers, and Bryan, your point is that in many places it's hard to detect any IBD, meaningful IBD above background between any given pair of samples. So it's not a universal metric, and I didn't want to present it as such, but I'm just trying to think of things that transcend the level of the markers themselves that might help the portability of inferences across different marker sets because of all the pragmatic reasons to adapt local genotyping approaches.
Mm-hmm. Yeah.
So I'm new to all this, forgive me if the question's too simple, but in one of the slides, Ananias, maybe it was yours, where you were considering all the different things that contribute to variations and import and analysis and so on. You had mutations and you crossed it out, and then most of it was about recombination. Is that driven by the fact that we can only analyze so many SNPs, or is it about something known about the relative rates of?
I think that might have been my slide. And it was just making the point that the model that we're using for the transmission networks is not really modeling mutations, because mutations in most areas of the genome, with the subtelomeric regions accepted, do not really occur at the time scales that we're interested in looking at movement over the period of months or years. But recombination, unless you have pure monoclonal infection, is occurring at a very relevant time scale, which is a transmission cycle. And so that's why we're really utilizing the reshuffling of those. And it's not even just the reshuffling of haplotypic organisms, it's actually the reshuffling of the polyclonality of the infections within these individuals that we're taking advantage of as well.
Yeah, I'm just going to go home and think more about this. Partly in terms of whether we'll miss the most interesting things that are going on. But I guess you need deeper data in order to be able to do the comparisons and so on. We may have such data in some of our collections.
And clearly mutations are important for parasite phenotypes and other things that we're very interested in, but less relevant or less need to necessarily include them for epidemiologic things where we just care about movement of parasites per se.
Yeah, so the South American study where you had very well controlled cases and the controls, hoping you even control the immunology, is there any indication that there are certain genotypes that actually will cause symptomatic infections and others do not? And if that's true, why are they not causing symptoms in the original person they are, from their cryptic transmission that's occurring? Any indication of whether we can start getting to know whether they are actually virulent strains and things like that?
Well, technically, the whole idea of the study, or what we have been doing over the years, is that trying to use relatively straightforward population genetic studies, but in very particular epidemiological settings when the controls and the cases are well defined. Because something that we found quite often is people, for example, comparing asymptomatics versus symptomatics and multiplicity of infection, but then when you read the paper, the asymptomatics happen in June and May in this village, and then the symptomatics happen in July and August in this other village. So at the end, you are comparing all the time apples and oranges. So in this case, what we tried to do, because the context was multiplicity of infection, was to carefully match complicated malaria cases or severe cases versus uncomplicated malaria cases, and then we match it on time and space, as you will do in any standard epidemiological study. And we simply try to rule out that there was some particular property of the genotypes that were related with that. And then you will find that the complicated and the complicated haplotypes are the same, and in the case of Peru, exactly the same. So you have longitudinal studies where you're going on the same communities, you have a clear case definition of a malaria case, you have asymptomatic cases, and when you do the genotyping, you find that all your symptomatic cases relate with asymptomatic cases, but then you find haplotypes in asymptomatic cases that you have not seen in symptomatic cases, which simply tell you, or at least that there is cryptic transmission that you are not seeing in the symptomatic cases, at least during that period of sampling that was 15 months, I believe. So 15 months is a lot of time to be asymptomatic, by the way. So you should see the same symptomatics and asymptomatics. So for me, 15 months was kind of the, wow, that's a lot of time to be asymptomatic.
So I wanted to bring, so I'm not a malariologist or whatever you even call yourselves. But I'm a human population geneticist, and so like one of the things I wanted to bring up was there's 2 points I wanted to bring up. One is IBD is great, but it also has basically an upper limit in terms of the number of recombination events until it breaks down beyond basically recognizability. And so in humans, we would say it's about 100 generations. You guys, I know that the generation times are all overlapping and it gets a little bit more wonky, but 100 recombination events is, in malaria is going to be pretty fast. And so, if you've got, we in this study that Shannon brought up, we cut out all of the people that were basically in the last 1 or 2 recombination events. The Tyler study, you guys were basically working with 1 recombination event, and so the number of markers could be far, far smaller for that scale of data than it's going to be for, say, 5 or 6 or 7 generations or recombination events ago. And so that's going to have to kind of play into part. The other thing is that I would bring up is a lot of the stuff that you guys are discussing are things that the human genetics community has had to deal with as well, in terms of developing what are called ancestry informative markers and those kinds of things, which is basically what you guys are doing with the microsatellites. And one way that we've come to kind of deal with that and kind of get an efficient SNP panel or a variation panel is to do ascertainment from different parts of the globe, and then to join that into one panel but then you know for each of the variants in that panel what its ascertainment was. So you know that this variant was found in, Oceania, and this variant was found in Southeast Asia, and this one was found in Africa, and how they might relate to each other. But then you have one universal set that you can use for everybody in the world. And then getting back to kind of some of the more pragmatic pieces, if as a community start to settle on certain technologies, the costs will start to go down. 100 microsatellites, that's what, I don't know what the cost is for that, but for some of these SNP panels that you could easily see with 5,000 or 10,000 markers on it, you're not talking about astronomical cost per sample, $25 to $50 a sample, if you can get the companies to bring the cost down.
Great.
Yeah, great comment.
Great comment.
Thank you.
So we're right on time. And unless there's any other.
There's one last little point, and one thing that maybe we can think about for next year is that one of our bottlenecks is analysis, getting the data genotyped, but then the analysis and the output. And one of the things that I think would be a great tool for our community would be some sort of portal where we could input genotyping data and it spits out interesting bits of information, such as geographic origin or drug resistance and so on. So that's something else I wanted to say.
Okay, well, thanks everyone, and have a good evening, and we'll see you in the next few days.
Okay.